Hello everyone, welcome. My name is June. In the next two modules, we're going to spend some time talking about population genetics. Before we start, I'd like to thank the sponsor for our program, the Department of Computational Medicine at UCLA, for supporting us for the website and also recording these videos. In the previous few modules, you probably have watched videos about the basics of genomics. So we have seen so far what DNA looks like, how we code different types of alleles, and how genes together with environment would contribute to how traits look like. However, so far we have been mostly focusing on the individual levels, which means, for example, in the Mendelian genetics, we talk about how these different types of alleles segregate and get assorted from parent individuals to offspring individuals. But if we zoom out of the picture a little bit, we just look at our context, most of the organisms on Earth don't live by themselves alone, but instead we live together with a group of individuals alike us. And we call this type of groups populations. So the way we define populations is a group of individuals of the same species, most of the time in the same geographical area, and we naturally have a chance to breed with each other. It should be noted that even though the definition of population is not hard, in the specific studies, the choice of population of how we define which individuals belong to one population can be actually tricky. So for example, take humans example, depending on your specific question you're asking, we can think population as a village, as a city, or even as an entire country. So that can be somewhat arbitrary, really depends on the specific question you're asking. So in population genetics, we study evolution over time. So it could be evolution in a short scale or in a large scale. But either way, we use population as the basic unit for these evolutionary forces to act upon. So just to clarify, for evolution, the basic unit is not individuals or species, but instead a population. So if we look at our human populations, there are two things that we can notice. First is we share a lot of things in common. We're not that different from each other on a high level. But then if we look close at each individual, we realize there's a huge amount of variations among us. There's no two individuals that are identical to each other. Even like the identical twins are different from each other in multiple ways. So in the 19th century, when people first started to study populations, one question people used to ask is how we actually maintain all this variation in the populations. And this is a question that confused Charles Darwin himself, that he couldn't really figure out, like if we have a population full of all sorts of individuals and we all mingle, we all mate, how in the world, after all these generations, we're still having variations? Instead, like, it's intuitive to think that if we started with a bunch of forks and spoons, we may end up with a lot of spork, but it is not really the case. So what is really going on with random mating and populations? So to answer that question, let's simplify the picture into as simple as a population with individuals that have only one gene, and this gene have only two copies, which we call two alleles. So let's call these two alleles big A and little a. So assume we have 10 individuals. For human and most of the diploid organisms, each individual will carry two copies of the same gene. It could be the same alleles or different alleles. So in this population, say we have 5 individuals in total. If each individual carry two copies of this gene, how many alleles are there in total? Anyone? 5 individuals, each one carry two copies. How many copies do we have in total? 10, right? Okay, so we call this, so if we forget about the entity of these individuals, we just look at these genetic materials people are carrying, we call this a gene pool. So this is a collection of all copies of all genes in the populations. It is essentially the same as thinking that every two of them is an individual. So if you still remember the concept of allele frequencies from previous modules, we call allele frequencies the proportion of each type of alleles in the population. So in this case, since we have two alleles, big A and little a, we can calculate the allele frequencies simply by counting how many big A's are there and how many little a's are there. Since we have 10 in total, we can simply count them as the frequency of big A being 0.6 and the frequency of little a being 0.4. So 60% versus 40%. And now let's look at what really happens with random mating. So we call random mating is literally we have two individuals, as long as they belong to two biological sex, they can have a chance to mate. They don't get to choose based on any other criteria. If we think about random mating, and if we think about this population being this collection of different types of genes, since we only have one gene in this population, and each of the individual would only get to pass down one copy of this gene. So random mating, or like we're producing one individual, it is essentially randomly drawing two alleles from this gene pool and make one individual. So if we have two types of alleles there, we can make a total of three types of genotypes. It's either big A, or big A, or little a. So what will happen with random mating in terms of what type of genotypes they can produce? So we can produce some big A individuals. And it's essentially drawing two alleles from this gene pool, and both of them being big A. So if we know that there are a total of 60% of big A's in this population, if we randomly draw two alleles, can anyone tell me what is the probability of me drawing two alleles of them both being big A?
Are we assuming we wouldn't be able to replace the allele after it's been chosen, right?
Well, let's assume this is a large enough population.
Okay.
Yeah.
So that would be just 60% squared, right?
Right. Right. So in the probability theory, if we're having two independent events happening together, we use multiplications. So since we have 60% of big A, drawing two alleles for them both be big A, the chance will be 0.6 times 0.6, which will get 0.36. Similarly, we can draw two alleles, and both of them being little a. So since we have 40% of them, we draw two alleles, and both of them being little a would be 0.4 times 0.4, which is 0.16. How about big A, little a? So big A, little a is a little trickier because there's actually two ways to get big A, little a. You can get a big A first and then a little a, which will be 0.6 times 0.4, or you can actually draw a little a first and a big A, both of them being the same thing. So we actually have 2 times 0.4 times 0.6, which is 0.48. Is everyone on board so far? Okay, so we have seen that with one round of random mating, we can still produce 3 different types of genotypes, and each of them have a specific chance of getting them. So now we're actually coming close to the fundamental principle of population genetics, which is called Hardy-Weinberg equilibrium.